Hi everybody, and welcome to our module on pyruvate dehydrogenase. Let's begin by talking about the potential fates of pyruvate. Pyruvate, as you know, is the end product of glycolysis, and there are a couple of options for where it can go once it has been synthesized by the process of glycolysis. As we're going to talk about in this module, it can be converted to acetyl-CoA by pyruvate dehydrogenase and then enter the TCA cycle. It can also be converted back into gluconeogenesis, and I talk about this process in the module on gluconeogenesis. There are also two other fates that you should know about for pyruvate. One includes conversion to lactate. This is often done by red cells, which lack mitochondria. It's often done sometimes in muscles, and then the red cells in muscles can release the lactate into the bloodstream where it can go back to the liver, and then the liver can convert it into glucose via gluconeogenesis, and this is called the Cori cycle. Another potential fate for pyruvate is conversion into the amino acid alanine. Once it's converted into alanine by muscles, it can carry nitrogen back to the liver. The liver can then extract that nitrogen and convert it to urea, and the liver can also convert the alanine back into glucose, which can then go back to muscle cells. And this is called the alanine cycle, which I talk about in more detail in some of the modules on amino acids. But for this module, what we're going to focus on is the conversion of pyruvate into acetyl-CoA, and whether pyruvate is going to be converted into acetyl-CoA via pyruvate dehydrogenase or go back into gluconeogenesis. For both of these processes, it needs to first enter the mitochondria. So let's talk about how it gets from the cytosol into the mitochondria. So as I said in the last slide, pyruvate is transported into the mitochondria for entry into the TCA cycle and also for gluconeogenesis. Don't forget that the first steps of gluconeogenesis occur inside the mitochondria, even though most of the process occurs in the cytosol. A brief review of the structure of mitochondria. This is a picture of one on the screen here. They have an outer membrane and then a space and then an inner membrane, and the inner membrane is folded into structures called cristae. But the point here is that pyruvate has to get into the center of the mitochondria in order to participate in the TCA cycle. First, pyruvate passes through the outer membrane through something called a voltage-gated porin complex, which is basically a channel that the pyruvate can move through. And then the inner membrane has a mitochondrial pyruvate carrier that moves pyruvate into the center of the mitochondria. Once pyruvate enters the mitochondria, it encounters two enzymes. It encounters pyruvate carboxylase, which is the first enzyme for gluconeogenesis, and it also encounters the pyruvate dehydrogenase complex, which is the enzyme that can convert pyruvate into acetyl-CoA for entry into the TCA cycle. Now, when ATP levels inside of a cell are relatively high, this will slow down the TCA cycle and levels of acetyl-CoA will rise. Acetyl-CoA is a very important activator of pyruvate carboxylase, so when levels of acetyl-CoA are high, pyruvate carboxylase will be activated and pyruvate will be preferentially driven towards gluconeogenesis. Acetyl-CoA can also inactivate the pyruvate dehydrogenase complex. This is a very smart system. It means that when acetyl-CoA levels are high, pyruvate will go this way towards gluconeogenesis, and when acetyl-CoA levels are low, it will go this way towards the TCA cycle. The pyruvate dehydrogenase complex is what converts pyruvate into acetyl-CoA, and it's called a complex because it's made up of three different enzymes. The first enzyme is called pyruvate dehydrogenase, and this is sometimes referred to as E1 because the complex is divided up into three subunits, E1, E2, and E3, like I've shown on the screen. And it is the E1 subunit that contains pyruvate dehydrogenase. The E2 subunit contains an enzyme called dihydrolipoyl transacetylase, and then the E3 subunit contains an enzyme called dihydrolipoyl dehydrogenase. This complex requires 5 different cofactors to function properly. We're going to talk about all of these in this module. The first one is NAD, the second one is FAD. It requires coenzyme A. It also requires the vitamin thiamine, and it also requires a substance called lipoic acid. Here is a picture of how the different elements of the pyruvate dehydrogenase work together to convert pyruvate into acetyl-CoA. The first step involves the E1 subunit. It adds thiamine pyrophosphate to pyruvate to release a molecule of CO2 and essentially add the carbon skeleton of pyruvate to thiamine pyrophosphate. Thiamine pyrophosphate is the active form of the vitamin thiamine. The next step involves taking that thiamine pyrophosphate carbon complex and combining it with lipoic acid to create a carbon skeleton that is attached to lipoic acid, and this is done by the E2 subunit. This lipoic acid structure can then be added to coenzyme A, and this will create acetyl-CoA. The purpose of the E3 subunit is to use the NAD and FAD cofactors in order to keep lipoic acid in the proper form to be used by the E2 subunit. So the net result of this entire complex process is that a molecule of pyruvate is converted into a molecule of acetyl-CoA. So now let's talk about those cofactors for the PDH complex in a little more detail. The first one we'll discuss is thiamine. Thiamine is also called vitamin B1, and it's converted to an active form of the vitamin called thiamine pyrophosphate, or TPP, in the body. If you look down at the bottom of the screen on the left side, I've shown you the chemical structure of thiamine. What the body does is take a molecule of ATP and remove two phosphate groups to convert the ATP to AMP. In doing so, it adds those two phosphate groups to thiamine and thus creates a molecule of thiamine pyrophosphate, which is the active form of the vitamin. Thiamine is a cofactor for four enzymes. The first one is pyruvate dehydrogenase, which we're talking about now. It is also a cofactor for alpha-ketoglutarate dehydrogenase, which is part of the TCA cycle, and also for alpha-ketoacid dehydrogenase, which is part of the metabolism of branched-chain amino acids. All three of these enzymes are part of a large multi-enzyme complex, just like the pyruvate dehydrogenase complex. And the complexes that use these three enzymes all employ the same five cofactors as the pyruvate dehydrogenase complex. Then the fourth enzyme that requires thiamine is a little bit different. It's called transketolase. It's part of the HMP shunt. It is not part of a multi-enzyme complex and does not require the five cofactors that the pyruvate dehydrogenase complex requires. It only requires thiamine as a cofactor. If you're deficient in thiamine, you cannot produce ATP because, as you can imagine, you cannot convert pyruvate into acetyl-CoA to enter the TCA cycle and produce ATP. The tissues that are affected most are those that are very aerobic and use a lot of oxygen, so this tends to be the nerves and the heart. The syndrome of thiamine deficiency is called beriberi, and it occurs in underdeveloped countries where children are malnourished, like this child in the picture on the screen here. The symptoms of beriberi are divided into dry symptoms and wet symptoms. The dry symptoms are polyneuritis, so this will be pain in nerves, also muscle weakness, and the wet symptoms come from heart failure, so the heart can get tachycardic and there can be high output heart failure and children can develop edema in the lower extremities. The other syndrome that occurs from thiamine deficiency is called Wernicke-Korsakoff syndrome. This occurs in alcoholics who are often malnourished, and in addition, they poorly absorb vitamins, so they can become thiamine deficient. This can lead to a characteristic cognitive syndrome where they are confused and they confabulate and make up stories, and I talk about the Wernicke-Korsakoff syndrome in more detail in some of the neurology modules. There's an important relationship between thiamine and glucose that you need to understand. If a patient presents to an emergency room in a malnourished state, for example, an alcoholic, they may be hypoglycemic, but in addition, they may have an underlying thiamine deficiency. If you administer glucose first to treat the hypoglycemia without giving thiamine, they will be unable to metabolize the glucose. If we go back a few slides, the glucose will lead to production of pyruvate, but without giving them thiamine, they will be unable to convert the pyruvate into acetyl-CoA. There are case reports of worsening Wernicke-Korsakoff syndrome among alcoholics who were treated with glucose for hypoglycemia without administering thiamine. So for this reason, you will often be taught when you do emergency medicine rotations that you need to give thiamine before you give glucose to patients who are malnourished. Now, I should point out that this practice has been criticized. There are also case reports of hypoglycemic patients having a delay in receiving glucose while someone looks through the cabinets to find the thiamine to administer, and while they're waiting for the thiamine, they have a hypoglycemic seizure. So many places are now practicing the administration of glucose immediately to hypoglycemic patients and then administration of thiamine as soon as it can be found. But you should know about this relationship for step one. They will often ask you why thiamine must be administered before glucose in patients who are malnourished. The next cofactor for the pyruvate dehydrogenase complex is FAD. FAD stands for flavin adenine dinucleotide, and this is a molecule that is synthesized from riboflavin, which is vitamin B2. This is the structure of riboflavin on the bottom left side of the screen here. On the right side of the screen is the structure of flavin adenine dinucleotide, and it is a molecule of adenosine. This is adenosine right here, added to riboflavin. This is the riboflavin structure right here. And since the word flavin is in the name of the molecule, it's easy to remember that riboflavin is required to create this cofactor. If you look at the top part of the screen here, I've shown you how FAD plus can accept two electrons and carry them as FAD. This double bond right here in riboflavin can be eliminated by the addition of two hydrogens. You can see it's not present over here. And once those two hydrogens have been added, the structure is carrying two electrons, and thus FADH is a carrier of electrons. And as we will discuss in the electron transport module, this FADH structure can also be used to generate ATP via oxidative phosphorylation. The next PDH cofactor is NAD. NAD stands for nicotinamide adenine dinucleotide, as I've written on the screen here. And just like FAD, NAD can carry electrons as NADH. It's synthesized from the vitamin niacin, which is also called vitamin B3. This is the structure of niacin on the screen here. You can get niacin from your diet, but can also be synthesized by the body from the amino acid tryptophan. And just like FADH2, NADH can be used in electron transport in addition to its role as a cofactor for PDH. The next PDH cofactor is coenzyme A, and just like NAD and FAD, this is also a nucleotide coenzyme. All of these cofactors, NAD, FAD, and coenzyme A, are synthesized by adding a vitamin to a nucleotide. In the case of coenzyme A, the vitamin is pantothenic acid, which is also called vitamin B5. This is the structure of pantothenic acid on the left side of the screen here, and this is coenzyme A on the right. And you can see that the pantothenic acid moiety, which is out here, has been added to a nucleotide right here to create coenzyme A. The utility of coenzyme A is for accepting or donating acyl groups. And the most important example of this is acetyl-CoA. This is the structure of acetyl-CoA on the screen here. And you can see it as this little 2-carbon group here hanging off the end of a sulfur molecule. And acetyl-CoA is going to give up this 2-carbon acyl group to other structures, and that's how it's used in the TCA cycle. It's also how it's used in the PDH complex process. I just want to pause here and mention that the first 4 B vitamins are all used in the pyruvate dehydrogenase complex. As we have seen, B1 thiamine, B2 riboflavin, B3 niacin, and B5 pantothenic acid are all cofactors for the pyruvate dehydrogenase complex. There is no vitamin B4, and thus the first 4 B vitamins are all part of this complex. They are all water soluble, and they all wash out quickly from the body. They're not stored in the liver like vitamin B12. The next PDH cofactor is lipoic acid. This is the structure of lipoic acid on the screen here. Lipoic acid binds with the amino acid lysine and forms a structure called lipoamide, and this is a cofactor for the E2 subunit of the PDH complex. An important thing for you to know about lipoic acid is that it is inhibited by arsenic. Arsenic is technically a metal, but it can be used as a poison by binding to lipoic acid. When it binds to lipoic acid, it inhibits the pyruvate dehydrogenase complex. Basically, it acts like a thiamine deficiency. When people have been poisoned with arsenic, they have a characteristic garlic smell on their breath, and that's because arsenic gets oxidized to arsenous oxide, which you can smell when someone breathes. In addition, there are non-specific symptoms from arsenic poisoning, including vomiting and diarrhea, and ultimately you can have coma and death. This is a picture of Napoleon Bonaparte on the screen here. You may know he was the emperor of France, and there is a theory that he was poisoned by arsenic. Arsenic is toxic to microorganisms, so it slows down the breakdown of tissue. And when they exhumed Napoleon's body, it was relatively preserved. So some people believe he may have been poisoned by arsenic. In addition, there have been some findings of arsenic in his hair follicles. But whether that's true or not, I always remember Napoleon as an example of how arsenic is toxic to the body, and you can remember that it's toxic through binding to lipoic acid. Pyruvate dehydrogenase is regulated through phosphorylation. There is a kinase enzyme which phosphorylates PDH and makes it inactive. There's also a phosphatase enzyme which dephosphorylates the enzyme and makes it active. I've drawn a picture of it on the screen here. On the left side in green is the active form. On the right side is the inactive form, which has phosphate attached. I've drawn some of the things which can trigger it to become active or inactive, and you can memorize this if you wish, but they make sense if you think about them intuitively. When there is a high level of NAD relative to NADH, that will activate the enzyme because by synthesizing acetyl-CoA, NADH can be synthesized by the TCA cycle. When there's a high level of ADP, this means the cell needs energy, so that will also activate the enzyme so that acetyl-CoA can enter the TCA cycle. And then finally, calcium is an important activator, especially for skeletal muscle, so that when you begin exercising, this enzyme will become active and feed into the TCA cycle. Inactivators of the enzyme are written at the bottom here. When you have a low level of NAD relative to NADH, this tends to inactivate the process for the opposite reasons I described above. When you have a high level of acetyl-CoA, this inactivates the enzyme. This is feedback inhibition, so that you don't have molecules going into the TCA cycle at the same time as they're going into gluconeogenesis. And then high ATP levels will also inactivate the enzyme so that more ATP won't be generated via the TCA cycle. There is a rare inborn error of metabolism called a PDH complex deficiency. Babies who are born deficient in the enzymes of the complex cannot effectively convert pyruvate into acetyl-CoA, and thus it will be shunted to alanine and lactate. If we go back a few slides here, babies who are deficient in the PDH complex cannot effectively convert pyruvate into acetyl-CoA. When the acetyl-CoA level is low, this inhibits gluconeogenesis, so this pathway is blocked as well. As a result, pyruvate will be converted into lactate and also into alanine, And so these babies are born with high lactate levels, and that gives them a lactic acidosis, which can make them very sick. This condition is often X-linked and occurs more commonly in boys. The most common cause is a mutation of a gene called PDHA1, and this codes for the E1 alpha subunit of the complex. So the key findings of a baby born with this deficiency will be poor feeding, growth failure, developmental delays, and on lab work you will see elevated levels of alanine and a lactic acidosis. Pyruvate dehydrogenase complex deficiency is really just one example of a number of very rare disorders called mitochondrial disorders. These are all inborn errors of metabolism, and they all cause severe lactic acidosis. When you cannot metabolize pyruvate inside of the mitochondria via oxidative phosphorylation for any reason, the pyruvate will get shunted towards lactic acid formation, and thus these babies will have a lactic acidosis. Key examples are the pyruvate dehydrogenase complex deficiency, which we're talking about now. You can also see a severe lactic acidosis in a pyruvate carboxylase deficiency, and there are also very rare deficiencies of some of the electron transport chain elements like cytochrome oxidase. The treatment of babies with mitochondrial disorders is very complex, but for step one, they like you to know some of the underlying biochemistry. So if a baby has a PDH complex deficiency, one of the first things you want to do is supplement their diet with thiamine and lipoic acid to try and optimize any functional PDH complexes that the baby has. In addition, you want to feed them what's called a ketogenic diet. So what's a ketogenic diet? Well, first of all, it has very low carbohydrates. You want to not feed them things that will be converted into pyruvate and thus into lactate. By feeding them low levels of carbohydrates, you will reduce the lactic acidosis. In addition, you want to give them a high fat diet. Fats can be converted into acetyl-CoA, which can then be fed into the TCA cycle without going through pyruvate. You also want to give them ketogenic amino acids. We'll talk more about this on the next slide, but lysine and leucine are called ketogenic amino acids, meaning they cannot be converted into glucose via gluconeogenesis, and therefore they can't worsen the lactic acidosis. And all of these things here are going to drive ketone production instead of glucose, which is what you want for the babies. You don't want to create glucose and pyruvate, which can then be converted into lactic acid. So leucine and lysine are the two ketogenic amino acids. They are metabolized into acetoacetate, which is a ketone body, and also to acetyl-CoA, which can enter the TCA cycle. But importantly, acetyl-CoA cannot be used for gluconeogenesis, and thus it cannot be converted back into pyruvate and worsen the lactic acidosis in babies with this condition. And that concludes our module on the pyruvate dehydrogenase complex.